So, welcome to know about our
இல்லை நான்
UIT
உன்னோட
course.
கனெக்ட் பண்ணல ரெண்டையுமே
Working on the UIT.
கொஞ்சம் இதில்
That's the
அட்மின் மட்டும்
UIT
வரும்.
website, web page. It's a home page for our overall process, it's including the publication, scientific blog, and the details about us, who we are, and what are the programs we are doing here in this Biotechnology Institute.
இப்போதைக்கு ஓரட்டும்.
First one is our chairman, Mr. Dr. A.S. Ganesh Raja, who did a PhD in Pondicherry University and he completed his postdoc fellowship in Dalian Institute of Science in China. After that, right now he is a assistant professor in National College Trichy. And myself, Dr. Rajkumar, who did a PhD in Pondicherry University, and I worked as a guest faculty in Central Institute of Tamil Nadu, Tiruvarur. And after that, right now I am a director of Regeneration Educational Trust in Viluppuram, and, and Dr. S. Chandrasekhar, who is a treasurer, who did a PhD in IIT Madras under the Professor Selva Segar, and he did a postdoctoral fellowship in Shanghai. Right now, he is working as a research scientist in Synergy company. And these are the three people who worked with us as a coordinator. Dr. Y. Sasikumar, who did his PhD in Anna University. Right now, he is working in China as a postdoctoral fellowship. And Dr. S. Tirumurugan, who did a PhD in Pondicherry University and who did a PhD in Pondicherry University. And right now, he is working as a Ashram professor in National College Trichy.
ஃபாஸ்ட்டாக
Third
பண்ணி
person
தொலைய
is
மாட்டேன்.
Dr. K.
மறுபடியும்
Vaitinagan, who did a PhD in Hong Kong. After that, he is right now in UK. He's a postdoctoral fellowship fellow. These are the overall framework of our UIT trust. We are doing the webinar series and UIT the TQs, scientific blog news, concurrent scenarios, and scientific scorecard and publisher and act as a publishers and YouTube channel we are running and scientific supports based on the requirements. Initially, we started with this scientific news blog, which is talking about the concurrent news with the various fields, not only in the science, not only in the chemistry or physics. We are spread out in the overall uh, blogs,
நாட்
which
ஓன்லி
is
சயின்ஸ்.
contain more than 100 EP contents. After that, we started a quiz program in chemical science, physical science, and also in mathematical science with the various experts. That's the first webinar series. We started with the Dr. K.T. Varghese, who is a director of Conclave, Bangalore. He talked about the patent rights, and Dr. L.R. Shobin, Assistant Professor in SRM University. He talked about the one-dimensional material and the perspectives. Third, by Balaji Thiru, about the robotic research and future directions. Fourth by Vaitinagan, who is a coordinator, sustainability through thermoelectrics. Fifth is a microplastic are everywhere by Chingam John. And sixth webinar by Prince J. Babu about the drug development challenges. And seventh is a
வாங்க
webinar
முடியும்னா வாங்கு.
by Professor Ratnesh Das from Harising Gora Central University, Madhya Pradesh. And eighth is the CSR
ஃபாஸ்ட்டாக
oriented
போக மாட்டேன்.
talk by
வேற
Dr. G. Rajkumar. And sixth is which
9th
is based on the instrumentation, the design
என்ன
and
பேசுனே
development of EEM water electrolyzer by M. Shankar Rao. That's the person who is going to give a talk about the molecular mechanism of regenerations. That's a today. And upcoming is by Leah Jacob, turning waste to products and carbon capture using industrial waste is that's the overall homepage of our UIT. You can be able to see what are all the programs we are running in online, which is uploaded after a certain period, so you can able to check. So this the call for book chapters. We are doing these book chapters also. The two reviewers is a faculty from Pondicherry University, Professor Anbalagan, and the eminent person from the University of Madras, Dr. T.S.M. Sangar Narayanan. Audible, right? So these are various chapters. So we can also collaborate with the Manonmaniam institution about the CSAR exams, call for scientific experts. So there's a doing process. We can also calling for the various scientific experts about the article discussions. So we hope we will do that one in future. So those who are interested in the content writing, just approach us for the scientific news. In addition to that one, we are happy to say upcoming sustainable chemistry and renewable energy virtual conference on the February month. So we are approaching the various institution professors and the scientists from the reputed CSR lab and the research institutions to give a invited talk as well as a advisory committee member for our overall process. So for that one, up to date, we are receiving the acceptance from the five members, Ms. Dr. Saravana Pichai from IIT Dhanbad, Dr. Venkataraman Magalingam from the ISER, Kolkata, and SS Bhattacharya, who is a site head, and metallurgy department from IIT Madras. And fourth one is Dr. Venkata Krishnan, who is assistant professor in IIT Mandi, and Dr. S.M. Jha, who is a scientific officer in Bharat as well as Rajaraman Center for Advanced Science from Indoor. So we are collecting the peoples for the for the completion of this overall seminars, and laterly we will inform to you also. Addition to that, everything is supported by the volunteers, this list of volunteers. Without these people, we can't able to complete this overall program. So once again, I would like to thank one and all departments for science and scientific activity. Hello, Sudha.
Thank you, sir.
Yes.
Every every person has the light with him. Every ray of light that is radiated by him. Make him shine in the wide world. Now I invite Dr. K. Vaidyanathan, coordinator of UIT, did his PhD in Hong Kong City University, Hong Kong, and currently working as postdoctoral at research assistant at James Watt School of Engineering, University of Glasgow at UK. For the introduction of chief guest.
Dr. Vaityanathan, your audio is. Please check your audio, please.
Am I audible?
Yeah, yeah.
Okay. I'm honored to introduce today's present. Am I still audible?
Yes, yes, audible.
Okay. I'm honored to introduce today's speaker, Professor Sudhakar Sivasubramaniam from Department of Biotechnology in the reputed Manonmaniam Sundaranar University, Tirunelveli, Tamil Nadu, India. His topic will be on molecular mechanism of regeneration. To to introduce about Professor Sudhakar's experience and Professor Sudhakar's experience. Oh, sorry, there is a problem. Yeah, Professor Sudhakar's qualification and experience is he graduated BSc from Madurai Kamarajar University and MSc from the same Madurai Kamarajar University in 1992 and PhD from same Madurai Kamarajar University in 1999 and he joined as a senior research fellow in the same university in Madurai Kamarajar University and he also worked as a postdoctoral fellow and the research associate at the UCLA at University of California, Los Angeles, from the period 1999 to 2007. And after his work at University of California, he also joined the same Manonmaniam Sundaranar University as the head and associate professor, and he's still as a professor in Manonmaniam Sundaranar University currently. And to tell about his memberships, he has two memberships from the reputed group of DNA Repair Group, NIH USA, and Cell and Molecular Biology Society. And about his funding details, he has a lot of funding from the government of India, which accounts to about 21 million Indian rupees till date, and he has successfully completed six projects, mm, two from Department of Biotechnology, two from the University Grand Council, and one from Tamil Nadu State Council in Science and Technology. And apart from this, he has successfully guided 12 PhD students to complete their doctoral thesis, and he has also awarded prestigious awards like the Bharat Siksha Award from the Art of Giving Charitable Trust and Most Inspiring Award in 2021. Most Inspiring Faculty Award in 2021. And I'm really honored to introduce Professor Sudhakar Subramaniam to give a talk on molecular mechanism and of regeneration. And about the topic, we are all interested to know about how the earthworm, when you cut the earthworm into two pieces, and it grows back again. And we are really interested to hear from Professor and the mechanism involved in the growth. I leave it to Professor Stager to continue. Thank you, everyone. Yeah, Professor Stager, can you please?
Yeah, can you hear me?
Yeah,
Yes,
we
yes.
hear you clearly.
Yes. Um, yeah, thank you for the very generous introduction. And uh, yeah, people around world are with us today. Around 50 people are joined together. Before starting the presentation, I would like to thank Universal Intellectual Trust, Viluppuram. This is very unique to see such a trust to augment the temper of science in the society. They are writing a lot. They have many blogs, and they are, it's they are bringing science to people. That we have to appreciate. Actually, if you see the foreign countries, they are giving equal weightage for teaching and also, uh, the service like this. University professors nowadays. mostly are teaching. They are inside the campus, but they are not coming out. But foreign countries, it is not possible. They have to do some outreach program like this. So this very much important. So I appreciate this trust in Universal Intellectual Trust from Wilbram. And also, writing is a big task. They are doing it. They are inviting variety of researchers. Uh, they are inviting a variety of researchers to share their knowledge among the society. That's also very important to stimulate the young minds for research and higher studies. So, uh, you know, and also they are doing a lot more, giving training for students for net and SLED examination. That also much important. So it's a pure service mediated trust. I really my heartfelt thanks to the committee, Dr. A.S. Ganesh Raja, the chairman of UIT, and also the secretary, Dr. R. Rajkumar of UIT, and Vaidyanathan, the coordinator of UIT, and all other members, the treasurer, Dr. S. Chandrasekhar of UIT. I really thank and congratulate all of them for their wonderful service for the society. Yeah, today we are going to see something about worm, earthworm. What do you see here um, in this slide is a worm. There are around 4,500 earthworms around the world. So many worms. Okay, so color-wise, they are very different. See here, the brown one is in kind of a man. It's called Dravida. This is from our Ghats, one among the biggest worm. Okay, and very colorful worms. Violet color worms, and also yellow color, and some worm from Australia is close to 75 uh, gram, close to a kg weight. Very big worms are there in Australia. In contrast, close to the North Pole, like 2 centimeter size worms also available. Very tiny worm also available. So likewise, the variety of the worm in the world is so high, around 4,500 worms. Okay, see here, very colors: blue color, green, and brown, pure white, and uh, you know like cement color. So many varieties of worms available um, in the world. So here we are using the worm called Udellus eugenia. Udellus eugenia is a um, African earthworm actually. Origin is Africa, and it's available all around the world. We can see around the world, but we are calling it as African earthworm. We are working using the worm for last 14 years. The reason is very easy to culture the worm. Okay, no much water necessary, and not much money necessary, and also very easy, no cost technology to rear the worm. So even without much money, we can do research. Only thing, for the fine chemicals we need money. Okay, and we have a lot of manpower. That's our uh, you know that's our students population, where we can use it, and it's easy for us to work with the worm nicely. Okay, and see here, now I enter into the topic. Actually, there are two different types of cells in the world. One is eukaryotic cell, another one is prokaryotic cell. Eukaryotic cell means, our cells. It has a nucleus, and it has mitochondria and so on. That's eukaryotic cell. But prokaryotes are, you know, bacterial cells, you know, they don't have nucleus. There's a single cell organism. Okay, there are two different types. So for eukaryotic cell, whatever the animal may be, the eukaryotic cells, they face aging problem. Okay, slowly, we are facing aging problem, and after death, slowly we are aged, and then finally we have to say bye bye. But prokaryotes is not like that. Prokaryotes, there is no aging problem. Always 18 years old. They are always young. Okay, and they actually the population is huge on the globe. Okay, and if you see the eukaryotic cells, there are two different types actually. One is plant cell, another is animal cell. You know, plant cell very unique. All cells are totipotent stem cell. Okay, in the plant, from a single cell, it's possible to generate the plant. That's the beauty of plant cells. But in the animal, it's not like that. You know, if we see our cells, our egg can fuse with sperm, and zygote is formed. Zygote, go to merula stage in in a couple of days. Go to merula stage. At that stage, the cells are totipotent. Okay, then the cells goes to the blastocyst stage, where the blastocyst stage where the cell lost the totipotency, and now it's pluripotent. Totipotent means entire entire organism can be generated. That's a totipotency. From a single cell, entire whole lot animal can be generated. In that case, we are using the term totipotency. Okay. Pluripotency means entire organism can't be generated, but yeah, organ can be generated. Many organ can be generated. That is called as pluripotent. Okay, so this is our body. Very begin with we have totipotent, and later our cells are pluripotent, and then after complete development in adult body, we have unipotent cell. We don't have totipotent cell in our body. Okay, so this is about capacity of our cells. So see here, this is the worm, Udellus Eugenia. So the mouth is here. Um, I can go to full screen. Okay, so the mouth is here, and the anus you can see here. So in between there is some area. This is called clitellum, a very pale buffy area. Okay, this is the clitellum starts from the segment 13 to 17. The five segments are called as clitellum. So if you open the worm, you can see variety of organ in it. For example, third segment we can see the brain, and then seventh segment we can see heart. Seventh, there is one eighth, and like the five pairs of hearts, the worm has it. In addition, ovary, testes, and so many organs are there in the anterior region of the worm. Anterior region means the segment before the clitellum. We are calling it as anterior segment. Then clitellar. Then the remaining body. From here to the tail is a posterior segment. That's what we are distinguish the segments of the worm. So it's almost like a train. The worm is a segmental animal. Each segment has a different organ in it. Okay, so it's almost like a train. Train has a different compartment. Likewise, here the worm has a different organ in different segments. Okay, so this is the fine detail about the segments and also the organ which are present in the worm. So here I introduced two different worms. Okay, one is Udellus eugenia. I have shown here. Suppose if you cut the worm just before the clitellum, okay, the small piece it dies, and the large piece, they regenerate. The entire body within 24 days. And if you cut the posterior region somewhere here, if you cut it, the small piece with the clitellum, the small piece with the clitellum, that regenerate. Okay, but the large piece, detached from the clitellum, it dies. Okay, and if you cut the middle of the clitellum, both the segment, the regeneration is started, but eventually they are not successful. Both of them are dying. So we found out that the clitellum is much important for the worm regeneration. Okay, that is sure, we confirmed. Okay, then you see this one is Perinois excavatus. This is actually Indian earthworm. Origin it is believed that Himalaya. So the worm is much different from the Utilis Eugenia in the view of regeneration. See here, if you cut in front of clitellum, the small piece it dies, and the large piece, it regenerates successfully. And if you cut at the clitellum, okay, the small piece dies, and the large piece it regenerates. Okay, and after clitellum, even if you cut in 10, five different pieces or 10 different pieces, no problem. It can generate so many worms. If you cut it 10 times, you can get 10 worms. So likewise, if you see the both the worm, uh, the Prenois excavatus is a totipotent worm, and here Utilis Eugenia is a pluripotent worm. We are working with a pluripotent worm because our body, is, pluripotency we have in our body. We don't have totipotency in the adult body. So it mimics mostly the human. So we are working with Eudilus eugenia to understand the regeneration issues. To understand the regeneration of the worm, first we have to understand the you know the skin of the worm. The skin, there are three different layer of cells. One is epithelial cell that is shown with the E, it's epithelial cell. Then is a circular cell is shown. Yeah, the circular cell, it's shown with C. Then it's a longitudinal cell. Longitudinal cell is really long cells. It's a third layer. First layer epithelial, second layer circular cells, and third layer is a longitudinal cells. So what you see here is a cross section. This is a longitudinal section. Here you can see the thin lines of all three cell layers. Okay, so it's almost like a cross section is almost like our chips, and this is almost like our budgie. We are angle of taking section is much different. It's a vertical, it's a horizontal section. Okay, and what you see here is tomb of ecom skin. Uh, so the first layer of cell epithelial layer, then second layer is circular, and longitudinal is the third layer. Then the intestine, and then this is about the bone. Okay, right. See here, we did first experiment. We took six worms. For three worms, we injected thymidine. Thymidine is a chemical; it can stop DNA synthesis. So by injecting thymidine, we stopped DNA synthesis. And this is the control worm. We injected plain water. Okay, and we cut the head of all six worms. After 3 days, if you see, the worm injected with the plain water, we can see the blastema formation. Blastema means new head. Okay, the new head is coming in the
போட்டுட்டியா?
control worm. Okay, uniformly we can see the growth of new head.
இதில்
But
வேணாம், இதில் கனெக்ட் பண்ணிக்கலாம்.
upon injection of thymidine, we couldn't see it. Okay, we couldn't see it. Okay, the new head is absent. That indicates, fresh cell division is important. If you want to see cell division, first the DNA should be copied. Okay, but
Windows
here
was
the DNA is
enabled to
not
connect.
copied because we stopped it by injecting thymidine, so DNA synthesis is not happening. Here the um, you know the regeneration also failed.
Hmm?
We did this experiment because there are some animals, you know, they regenerate even without mitosis. Like planaria and hydra, several animals, they
இதில்
are
தான் பண்ணுறேன்.
migrating the cells, their body
ஏதோ
cells, they are
ஆல்ரெடி இதில்
migrating the cells,
போயிட்டுருக்கனால கனெக்ட்
and the cells are
கனெக்ட்
differentiating,
ஆயிடுச்சு.
and then finally,
ஆயிடுச்சு.
you know, they can regenerate. So this is possible in some varieties of animals, but the worm, it's very important to know that the mitosis is much important for regeneration. Okay, see here, this is our experimental strategy. For example, we have a fine blade, and with that we are cutting the head. And after third day, we can see small head, new head is slowly growing. Then what we do, we are cutting the head, a section of tissue we are taking out, and the tissue we are looking at a microscope. Then we can see what's going on. Okay, this what we saw. Okay, just after cutting the worm, we are cutting the head. Just after 15 minutes, we saw the worm under microscope. This is the cutting site actually. This and this is the cutting site. Here, this and this is the cutting site. So what happened immediate after cutting the worm? Cells. No, cells are elongating. Like uh, 12, 15 times. Cells are really elongating, and then you know uh, they are migrating by means of elongation. They are migrating. Okay, the worm also migrating in the same way. They expand their body, then they're stringing, expanding and stringing. It's helpful for the worm to migrate. Similarly, the worm, cells within the body, they manage the wound healing the same pattern. They really expand their size. See here, all the cells are really, elongated. Very long, long cells we could see at the site of amputation. What you see here is that 15 minutes after amputation, okay, the long cells you can see, and here is a 30 minutes after amputation, still the cells are really elongated. Okay, then after 1 hour, the cells now is very different. Okay, they came to the normal size, and within 1 hour, they can heal the amputation site. See, it's remarkable. If what happened in our body, if something happened, small wound or some incision happened on our body, it takes at least a week to heal. But in this worm, within an hour, you know uh, the cutting site, the amputation site is completely healed. Okay, so for this, our data says no new cell division necessary. Even if you stop DNA synthesis, this wound healing is possible. So it's very nice. Simply the cells are migrating by elongating their body, and it can nicely heal the amputation site, the wound of the amputation site. It's nicely doing. Okay, so this very nice model to understand how the cells are migrating. How, what are the factors inside of cell migration. All those things we can study. Okay, so this was happening. Cells are elongating, and then it's migrating. Okay, that's the thing. And if you see the regeneration, okay, it's very different. For wound healing, the mechanism of cell behavior is much different. But for regeneration, the cell behavior is much different. In the case of regeneration, what happened? The cells from the longitudinal layer, the third layer, they are migrating, and they form the blastema. See here, this is the control bone. See, this is the clitellum actually, 13, 14, 15, 16 or clitellum. The clitellum, you can see a thick layer. Okay, this layer is called, granular epithelial layer. This layers of cells, the population of these cells are huge in the bone. And see here, this is the 10th segment, this is the 11th and the 12th. Here, if you see, the epithelial layer is just a thin line, and circular are also very thin line. And if you see the third layer, the longitudinal layer, also thin line. But after amputation, third day after, if you see the population of cells, they are much different. See here, very thin layer, the epithelial layer, no difference at all. Okay, and if you see here, the second layer, the circular layer, here in the regenerating bone is missing. It's all gone. Okay, it's all disappeared. And if you see the longitudinal layer, okay, now it's a dominating. So in this 10th segment, 11th and 10th, if you see there's the 11th. So epithelial is there, and then the longitudinal cells are there, and the population is more than 10 times higher. These cells, population increases, in the longitudinal cell population increases, and then they slowly migrate, and they form the blastema. Okay, the new head is is because of the longitudinal cells. Okay, now we can understand a little bit clearly. This is epithelial cell, this is circular, this is longitudinal. Just after cutting, the circular layer completely disappeared. There is no change in the epithelial. Circular layer completely disappeared, and the population of longitudinal cells are enormous. Okay, their population increased rapidly in 3 days of time, and those cells, they grow, they migrate, and they form the blastema. The new head is accumulation of the longitudinal cells. Okay, so this is what we understood, and now if you see, we have to think why the circular layers cells are disappearing. So what's the science behind it? Okay, that we have to understand. Then, you know how the longitudinal cells population increases. What's a background of it, we have to understand. That's the second step. Third step, how this longitudinal cells. But please remember, the new head has full of longitudinal cells. The longitudinal cells, has the ability to differentiate into varieties of other cells. See here, so many other type of cells. Epithelial cells, and also circular cells, and also the neuronal cells, and the muscular cells. So many other cell types are there. All should be formed from the longitudinal cells. So how it happens, that is very interesting aspect of the bone regeneration. To understand, you know, some background about worm regeneration, we use a technique called BRDU labeling retention assay. It's very easy. They are labeling the cells with a chemical called BRDU. BRDU means brominated uridine. What is brominated urine? If you ask a question, this is nothing but diamine. In DNA there are four different bases: guanine, adenine, thymine, and cytosine. Here the diamine we are taking, and now the diamine, we have modified the diamine with the addition of a bromine atom. So one bromine atom we added into diamine. Now the molecule is much different. We are calling it in a different name. Brominated uridine. Okay, that's a name. Simply you can remember as brominated diamine. Okay, so assume four molecule of brominated diamine. We are adding to a cell, just four molecule. We are adding it to the cell. Just you assume. Okay, so the cells took all the four molecules. All the four molecules are loaded in the DNA. Okay, so it's a DNA precursorate, so it can be added into the DNA molecule. So four molecules of the BRDU is loaded in the DNA. So now the cell is dividing. One cell now is two cell now after mitosis. So in each cell, one cell we can have two, BRD molecule. Other cell, there are two BRD molecule. After that cell division, this is possible. And now the two cells again they divide. If you assume now you have four cell stage, now there is a possibility. Here is one molecule of BRDU. Here is another molecule of BRDU. Likewise, each cell has one molecule of BRDU. This is this is the uh, uh, distribution of BRDU after second division of mitosis. Assume the cell is dividing again, the third division. Now you have eight cells. Now, can you detect BRDU? No, we can't detect the BRDU because it's diluted so much. So we can't detect BRDU anyway. Okay, so this is a technique. Okay, to understand which cells is dividing in our body. Okay, which cell is not dividing, we can use this to find out this. We can use this technique. For example, for example, the slow dividing cells, are stem cell. Mostly stem cells are not dividing; they are sleeping. Upon requirement, they divide. Exception is there. For example, our neuronal cell, they won't divide. Okay, they are poison cells. They won't divide at all. They stay as such for a long time. This is an exceptional case. Okay, mostly the cells in our body are dividing, except the neuronal cell and stem cell. The cells are dividing more. So using this technique, BRD retention assay, it's easy to find out the stem cells. And also there are many other way. We have marker protein, for example, stem cell marker are there. We can use that marker also to find out the stem cells. So variety of techniques are there. We used all and we found out the stem cells. Okay, the stem cells has been characterized in the worm, and we published already. Okay, so this is another experiment. Okay, we injected BRDU to the worm. Okay, we injected. After injecting, we left the worm without disturb. We didn't disturb the worm for 30 days. Just today we injected. We have 30 days. We have to wait, and 31st day we have to cut the head of the worm, and 34th day we can see small head slowly growing, and take the section of the small head, and then we have to process with antibody, and then the tissue we have to see under microscope, and we can get this data. Okay, this data says, see here, this is the newly developed head. Okay, this line, vertical line, is the amputation site. Here we cut the worm into two. This is the amputation site. The tissue, all the tissue, okay, is newly generated tissue. This is the new head. Okay, it was generated just 3 days ago. Within 3 days of time, the tissue was generated, and we checked the tissue, and we saw the BRD labeled cells are within this newly generated tissue. Okay, the brown color cells are BRD positive cells, or the BRD positive cells are in the newly generated tissue. The newly generated head, we can see those cells. That means, you know, the BRD cells should be in the body. Okay, the newly divided cells, they have BRDU. That means some BRD molecules having cells are migrating to the head. So those cells are, you know, stem cells. The stem cells are much important in that region. They can differentiate and they can divide, and they can generate organ. So this is the stem cell which are present in the bone. Okay. So this is the conclusion. The stem cells are here in the worm. They migrate, and they reach the newly forming head. That's a point the previous slide told you. Okay. So then we check the worm. Uh, uh, the skin of the worm, we checked it. The stem cell is in between the first layer, the epithelial layer, and also circular layer. In the boundary, we can see the stem cells. So upon amputation, suppose we cut the head, the cell population increases, several fold. In the control worm, there are few stem cells are there, but if you cut it, cut the worm, and slowly the stem cell population increases a lot. And in the uh, longitudinal cells, we can see more cells of stem cells, and they migrate and they form the new bed. This is what happening. So the stem cells are in circular layer, and upon amputation, they migrate to the longitudinal, and also their population increases in the longitudinal cell layer. Then those cells are migrating from there to the blastema. So the blastema has a lot of stem cells. Okay, so this is the point. Now we have to understand one more thing. Mostly the cells, if you see under microscope, you can't see it because it's almost transparent. Cells in our body, are mostly transparent. And if you want to cells, you want to see, you want to see the cells. There are two different methods. One, you have to stain the cells, and then you can see under microscope. Another method, you have to have stereoscope, stereo microscope. Okay, there, with the shadow technology, you can see the cells. So the first image, what you see here, is a stereo image. Okay, shadow techniques are used, and the cells were visualized. But here the cells were stained, and now you can see the cells. So there are two different methods to these cells to see the cells. Okay, but there are some exceptions. For example, our RBC, without staining, we can see it. Because the reason behind it is, the RBC has a lot of, you know, um, hemoglobin. Hemoglobin, the RBC has. The hemoglobin is red in color. So nicely we can see the cells. Okay. So in the one more cells, for example, our skin cells, especially our skin is little darker skin. Reason is melanin. Okay. So the skin cell melanocyte produce a lot of melanin. It's a black color chemical. Okay. So these two cells, without staining under microscope, we can see it. Okay. Other cell we can't. So assume, you have a technique, suck all the two dyes from our body. What are they? Melanin and also hemoglobin. If you suck these two chemical out of a body, the person will be invisible. The light easily can pass through the person, and the person can be invisible. Okay, so that's the thing. Okay, so now see here the other slide. This is to teach you. Okay, so see here the picture of Lord Krishna. Okay, that's a halo around his head. See here, this is a sun. There is a halo around sun. See here is a Jesus. We can see a halo around Jesus. So this is uh, Gandhi. We can see a halo. This is we drew it. Teresa also we have halo. We drew it. But none of the halo except the sun, we we can see it. Okay. But normal people, we don't have halo. Okay. See here the stem cells. Here is these are the stem cells. Okay, the arrow indicates stem cells. The stem cells under UV light, they glow. Normal cells, they are not glowing, they are not colorful, but stem cell, under UV, they are glowing. First, we noted in the worm, okay, the fluorescence property of stem cell in the worm. So the fluorescence is not in one color. The fluorescence is from red to blue. The fluorescence is wide range, broad range of fluorescence is there for the stem cells. See here, this is a stem cells. Here is very bright. Okay, that's the beauty of stem cells. It's almost, normal cell is not fluorescing, but stem cell is fluorescing. Okay, that's the thing. So we wanted to understand, you know, why the stem cells are fluorescing. Okay, in the same period, in the human body also, a scientist from US, he took the hair follicle stem cells. Okay, and he observed under microscope. He saw the cells are fluorescing. Stem cells are fluorescing in human also, and stem cells are fluorescing in earthworm also. So fluorescence of stem cell is somehow is conserved from earthworm to human. So, but at that time, the reason behind the fluorescence is not known. So, human stem cell harvesting and working with human stem cell very expensive, very time consuming. But handling of stem cell in the worm is relatively easy. So what we did, we cut the head of the earthworm, okay, and then we waited for 3 days. Within the 3 days, the stem cell populations are so enormous in the amputation site. We took the stem cells, okay, we harvested the stem cells and then we crushed it. We got the fluid and we checked under we checked using thin layer chromatography. We saw fluorescence. Okay, so what we did, the first plane is a riboflavin, okay, that is purchased from Sigma, and the second layer is a juice of earthworm. The amputation site tissue was taken, this was squeezed, we got the juice, and the juice was subjected to a TLC, and we saw a bright spot. Under UV, we saw bright spot. So the very bright spot of riboflavin and also the stem cell juice is almost, the mobility of these two spots are almost same. That indicates the stem cells also has rich amount of riboflavin. So what we did further, we mix these two chemicals, riboflavin and also the earthworm stem cell, juice. We mixed together and we ran one more slot, and we couldn't separate it. That means both may be the same chemical. So okay, to understand what we did a lot of spectroscopical experiment, and then we found out, yes, this stem cell is fluorescent because of riboflavin, that we confirmed. Okay, so another question arise, you know how the worm is having a lot of stems, a lot of riboflavin. Riboflavin is rich in stem cell. That's what the stem cells are glowing. Now we have to understand from where the worm is getting the riboflavin. For us, supplier of riboflavin is we are eating a lot of vegetables, a lot of fruits. Even we are buying the riboflavin from the shop, we are eating it. So there are three different methods we have riboflavin. But the worm, who will give rich amount of riboflavin? That's what we wanted to understand. For that, we injected worm with a good dose of antibiotics. Okay, very good dose of antibiotics. We injected the worm for several days. After that, we checked the presence of riboflavin. Okay, so this is earthworm injected with antibiotic. This is the control worm. In the control worm, riboflavin amount is very good, but in the antibiotic injected worm, the riboflavin concentration was really really low. Okay, around tenfold less of riboflavin in worm injected with injected with antibiotic. So the data says if you inject a good amount of antibiotic to the worm, the gut microflora and associated worm associated microflora are dying. So the microbes are dying, and eventually the production also reduced. So the result says, riboflavin is produced by the gut microbes. The body, worm body has a lot of microbes. That microbe, they produce the riboflavin. That's what the data suggests. Okay, then we characterized bacteria. Actually, we isolated a bacteria from the worm. Okay, this is a bacillus. Okay, the bacillus is also flexible rod. Okay, this bacteria produce good amount of riboflavin. I'm not saying this is the only bacteria can produce riboflavin, but I can say that this is also one among the best organism for production of riboflavin in the worm body. Okay, so this organism is much important for the worm for successful regeneration. Okay, so that's what we understood. Okay, this is the colony, and this is a microscopical observation of the bacteria, and this is the evidence to show that the bacteria can produce good amount of riboflavin. Okay, also the bacteria produce one more fluorescent molecule. We don't know what it is. Okay, so this is the thing, and this, one more data I want to show you. See here, third day worm. Okay, we can see small head on third day of amputation. We can see small head. Fifth day, the uh, newly generated head size is increasing. Okay, small head here on third day, it's now bigger in fifth day. On seventh day, it's still bigger. Okay, then what we did, we injected riboflavin in the regenerating worm. We injected riboflavin. See here the size of bud here, the newly generated head, very small here. But if you inject riboflavin, the size is much bigger. See here, and also note here, it's very big size of blastema. We can see upon injection of riboflavin. So you see here, seventh day, a very nice bud, but here is very long bud, we can see. So injection of riboflavin, that increase the speed of regeneration that we saw experimentally. Okay. So another thing we did, we injected antibiotic. We injected a mixture of antibiotics. See here, it's not, third day we couldn't see blastema, and fifth day is very small, and the seventh day the blastema red in color, and eventually the worms are died. So if you inject antibiotics, it population of riboflavin reduces, and regeneration also affected, and so regeneration is not possible, upon antibiotic injection in the absence of riboflavin. Okay, so here we mixed antibiotic and also riboflavin. Here there's a question comes. Antibiotics has a lot of side effects. Okay, so the failure of regeneration may be due to side effect of antibiotics. That may be also a reason. So to clear the question, we did another experiment. Antibiotics and riboflavin were mixed together and we injected, and nicely we can see the data. The bone regeneration is augmented, almost like the riboflavin injected worm. Here, antibiotics and riboflavin mixed together, if you inject, nicely we can see regeneration. So this what that we found out, and very useful, to understand biology of the stem cells. Okay, so this what going on. Okay, the worm gut has riboflavin producing organism. Okay, so if you cut this head, what happened? Worm can't eat it because there is no mouth, so worm can't eat it. Okay, so yeah, under that condition, the gut microbes also under stress. Gut microbes also can't eat it, so they are under starvation. Under starvation, they produce riboflavin. The gut microflora under starvation, they produce riboflavin, and that riboflavin was enjoyed by the worm. They suck in all the riboflavin and they use it for their regeneration. We found out this association, symbiotic association of bacteria and the worm by following experiments. You know, we took the microbe, we isolated from the bacteria, we took that microbe, and we grew it on a plain medium. Okay, it's a minimal media. The media doesn't have riboflavin. Okay, the organism slowly grows. Okay, the blue line is the vegetative cells of this bacteria, and the white line is the riboflavin concentration. Blue line is the bacterial concentration. White line is the riboflavin concentration. So the bacteria is slowly growing. What we call the flat line. The flat line is a lag phase. This sharp lane, is a log phase. This is a stationary phase of bacterial growth. So stationary phase, log phase, lag phase. The bacteria couldn't produce riboflavin very beginning. In the lag phase, there is no production of riboflavin. Also, in this lock phase, the bacteria is not producing riboflavin, but the riboflavin is produced by the bacteria only during the stationary phase. So, okay, that means in the lag phase, a lot of nutrition available. In the lock phase, also a lot of nutrition available. But in the stationary, the nutrition is depleted. The organism get all the nutrition. Now the bacteria produce riboflavin. That time it produce riboflavin. So based on this data, we drew this schematic diagram to inculcate the association of microbes and worm, okay, to the community. Okay, so this is the bacteria just in a day. After third day, the bacteria turned into yellow color. Okay, so the bacteria color is due to riboflavin. So very beginning is not producing riboflavin. If you incubate the bacteria for longer time, we can see the riboflavin production. Also at that time, the bacteria is not in a vegetative stage. Now it's in a spore forming stage. Okay, during the spore formation time, the bacteria is producing a lot of riboflavin. Bacteria is producing riboflavin because okay, the germination of spore needs riboflavin. That's why the bacteria is producing riboflavin. Okay, now see here, this is a worm skin, earthworm skin. All the bright spot are stem cells. Very sharp bright spots are stem cells. If you inject, a lot of antibiotic, the bright spots all gone. Okay, that's a problem. The worm, fails to regenerate. Okay, yes. Okay, this is about the stem cells. Okay, and now we saw just after in a control cell, in a control worm, for example, this is a septum. Worm has a different segment. Each segment is separated by a tissue called septum. This is the septum. Okay, in the septum there are some cells. Okay, this is a longitudinal cells. The longitudinal cells, a type of cells is hanging. Okay, see, here is the longitudinal cells. There a type of cell is hanging. We observed, okay, the cell behavior during regeneration. Okay, in 15 minutes of time, this hanging cells, the blue color cells, in the population is increasing a lot. Then we did a lot of other experiments, and we found out these cells, the blue color cells, are nothing but mast cells. Okay, mast cells. Okay, the mast cells is our body also has the mast cells. Okay, it is produced in blood, and then the cells are penetrating tissues, and it's living between the our tissues. Okay, tissue attached cells is a mast cell. So the population of the cell is increasing, you know uh, just after 15 minutes of amputation. Okay, and you see here, and 30 minutes, the mast cells releasing a lot of factors. Still we are studying what are they. They're releasing a lot of factors. Those factors, nicely, you know helpful to attract the stem cell to the point. So the mast cell is releasing a lot of factors, and those cells, that factors is attracted by, uh, you know, yeah, the chemicals attract the stem cell to the point. Okay, so see here, on third day of regeneration, so much mast cells. Okay, they are degranulating. They are releasing a lot of factors to attract, stem cell to the point. Okay, so that's the thing. So the mast cells in the earthworm body is also playing a major role, okay, in regeneration of the worm. Here I show you another data. So this is the worm, first segment, second segment, third segment, and fourth, fifth segment. Likewise, we can see segment by segment, we can see the worm. Here, the red color is nothing but, albumin. So albumin can be stained, sorry, collagen. not albumin, collagen. Okay, the red color one is a collagen. So collagen nicely can be stained. Okay, the collagen is rich in the uh, third layer of the skin. So third layer is having longitudinal cells. So longitudinal cells having a lot of collagen. So what is collagen? Collagen is a cement material. They hold the cells in a place. Okay, so a lot of cells are there in longitudinal, in the third layer. The cells are attached to each other by collagen. Okay, that's what the slide says. Okay, so the cells, see here nicely, the collagen is rich in the third layer. We cut the worm. Okay, we cut the worm. Okay, see, in after just one day, see here, collagen all gone. In the body segments of the worm, the collagen is missing. But all the collagen now we can see in the amputation site. So the collagen is migrating. Okay, also the collagen is missing in the third layer of the worm, regenerating worm. So what does it mean? you know During regeneration, the cells are migrating. Okay, and as for the migration, the cement material should be broken. The cement, what is cement material? The collagen is holding the cell. Those molecules should be broken. Then only the cell can move from one side to another. That's what happening nicely. The collagens are removed from the body segments, and they are coming now in the plastema. Now the cells in the third layer is movable, is motile now. The cells are moving, and nicely we can see the regeneration. This is of day one, day two, and day three. Nicely we can see the collagen reduction at the third layer of a tomboid. Okay, so that's what happening. See here, control cell. I told you before, the collateral is buffy and pale and buffy. But during regeneration, what happened? All this buffiness is completely gone. Okay, so why? Because the collagen in this region is completely removed on during regeneration. So all those cells, the blue color cells, during regeneration is completely migrated to the amputation site. Then the regeneration is possible. Okay, so this is the mechanism. The collagen degradation or dissociation, supports migration of cells from the third layer and also from the collateral. This for this cell migration, TCTP is another protein. Okay, translationally controlled tumor protein. TCTP means translationally controlled tumor protein. This protein is much important for the cell migration. That we proved it. Okay, so another method. See here. Suppose if you cut the worm, okay, in the anterior, okay, here right here, if you cut the worm, this stem cells from this clitellum is migrating to anterior side. If you cut the worm right here after clitellum, the stem cell from the clitellum is migrating to the posterior side. The slide nicely shows that. Okay, if you cut it here, the stem cells are migrating towards head. If you cut it, the the tail, the stem cells are migrating towards the tail region. That's what the data suggests. Okay, like this. If you cut it right here, the stem cells are moving to the amputation site. Suppose we cut it the other side, the stem cells are moving to the other side. That's what the slide is showing. Okay, so likewise, okay, we found out, okay, some background, some scientific evidences of how the worm is regenerated. See here, if you cut it here, the small piece dies. Okay, the large one survives and regenerates. And if you cut the tail region, the large piece dies, the small one regenerates. And if you cut the clitellum, both regenerates, but abnormal regeneration we can see, and worms are dying. That's what uh, uh, we saw. And the reason is the stem cells are in the clitellum. Stem cells are life-saving port for the worm. Okay, they have a rich amount of stem cells, and the cells saves the worm upon amputation and to regain the lost body parts. Okay, that's all from me. Okay, my lab also sequenced the entire genome of the worm Utilus genia. Also, we have annotated around 22,000 genes of Yersinia fetida, that's the Indian earthworm. Also, the whole genome we completed in our laboratory. We completed almost all gene RNA sequencing, mRNA sequencing also completed more than like 60,000 more. I'm saying all the splicing variant and all. So we have completed all the uh, RNA sequencing from the worm. Okay, also three four bacteria associated with the worm have been sequenced. Okay, by us. So all those sequencing data is so much important to understand cell biology and the genetics of the worm for better understanding of regeneration biology. Okay, so this is the conclusion. Stem cell is fluorescent. The fluorescent nature of the stem cells was first decoded in Tirunelveli by us. Okay, this reason is, okay, riboflavin is the reason for the stem cell fluorescence. The ribofavin is supplied by gut microbes. Okay, gut microbes, is important for the supply of ribofavin. Okay, then the stem cell migrates from one place to another. Okay, uh, that we improved it. Mast cells has a major role in stem cell migration. Stem cell migration is supported by collagen dissociation. Collagen molecule should be dissociated, then the stem cell can be migrated. And the stem cell niche, okay, there are two niche. One is the boundary between epithelial and circular cells is a stem cell niche. Another very big niche of a stem cell is clitellum. The granular epithelial cells of clitellum is another major source of stem cells. Okay, that's all from my side. I thank all my funding agency, DVT, DST, and ICMR, and so on, and also UGC. They gave a good amount of money initially to establish laboratory, also for students and fine chemicals. Okay, yeah. Thank you for listening. If you have any questions, I'll take it. Thank you so much.
Thank you very much for your nice presentation and nice talks. We are really excited about your talk. Some of participants posting some questions
Yeah,
in
your
chat
voice
box.
is so low.
Oh, okay. So some questions
Yeah, there are
in
some
chat
questions
box.
right
Yeah,
in.
yeah, yeah, yeah,
Yeah.
yeah.
In case where the head of worm is amputated along with blood lump, we saw regeneration, but it doesn't have in the stain. I couldn't understand. In case where the head part of ribofavin is amputated, okay. The usually we are amputating the worm in two different places in my laboratory. One is the tenth segment, that is the anterior region, tenth segment. For all experiments, we are amputating at 10 segments. And for the posterior side, we are amputating at 30th segment. It's much complex. The worm is much complex. Regeneration is already very complex process. To cut short the complexity, to study the posterior, we are cutting at 30th segment. And to study anterior, we are cutting at 10th segment. So that's what we are doing. Okay. Okay, next question. Will you please say the procedure of bromination of diamine? Uh, no need. Just chemicals are available. Sigma, they have it. Buy it. I don't know the chemistry behind the synthesis of brominated diamine. That is BRDU. Uh, just we are ordering it. Okay, the company Sigma and other company they have it. we can buy from them. Okay, we are not synthesizing the BRD chemical. Okay, because it's laborious. We need equipment for all those. We need expertise in that area. Then only that kind of work can be done. Simply, we can't change the direction. Okay, just we are buying and we are using it. Our major goal here is to understand regeneration. Okay. uh,
Dr.
yeah.
Varghese sir, you
So
have you can directly connect
yes,
with it.
yes,
If
in the
you
case
have
of
another
human
questions
being,
or doubts, you can
do we
directly
get
connect with.
riboflavin from bacteria or any other source? Of course, we also have a lot of microbes, beneficial microbes, lot of microbes in our gut. Okay, so they are producing good amount of riboflavin. Not only riboflavin, vitamin B12, B2, so many. Okay, all produced by our gut microflora, and we are enjoying it. So if you see, in case of while you are taking antibiotic, the doctor used to write vitamin also. So when you take antibiotic, you have to take vitamin also. Capsules, vitamin capsules also you have to take it. Reason, the intake of antibiotic will kill the intestinal and colon microflora. They are very great resource for supplier of riboflavin and other unknown very beneficial molecules. So in that case, our body will suffer. So we have to supplement with these vitamins. Okay, so in our case also the same. That's what I believe. Now, from bacteria to human, we all same. Our shape and size differ. Okay, our skill differs, but from bacteria to human, we all same. So if you do experiment with earthworm, we can understand a good amount of knowledge about human body, because the basic biomolecules, DNA, RNA, protein, lipid, carbohydrate, all same. Okay, from biochemically all same. But the sequence, the DNA sequence, there are some differences between organism to organism. So when we use any organism for by experiment, a certain percentage of human knowledge can be revealed. No need to directly take human sample for research. Please remember, most of the eye disease was found out by Drosophila. Most of the hearing related genes were found out by Drosophila. So Drosophila, what is Drosophila? Is a fly, fruit fly, very small, tiny fruit fly, helped us to understand lot about genetics of eye, lot about genetics of our ear. Okay, that's the beauty of biology. So understand a organism, and we can understand our body. Okay, like in a way, in a sentence, I can say around 80% our body and earthworm are same. Our cells and earthworm cells around 85% we are same. The difference is just 15%. So understand worm, 80% of human problem can be solved. Okay. So for example, earthworm doesn't have bone. So bone related work can't be done with the worm. Okay. So in the case of regeneration, it's amazing. A scientist told, okay, without regeneration, no organism can survive on the planet. Okay. With the complete regeneration, no organism will die. Okay, that also possible. Regeneration, if it is failed, no organism can survive. Also, if 100% regeneration is possible, no organism will die. So we are in between. We all organisms are in between. So understanding regeneration is ultimate goal for solving diseases and other body related disorders. Okay. Yes. Any other thing? Okay. In case of human being, we get this. I finished. Okay. Yes. Any other question?
Any other questions from participants, please? Please, you can unmute and ask. Okay, sir. I have one question. That
Yeah,
is not question, just like it some doubts, because I from chemistry background, so I am thinking about how this chemical fertilizer we are using some fertilizers in for agriculture field. How to affect regenerations of earthworm? Is it any possible to affect the regeneration of earthworm or
yeah, yeah.
something
Very,
else?
very nice, nice
Yeah,
question. I can tell you.
yeah.
You know, I used to trek. Okay, we have Western Ghats very close to us.
Okay.
I used to going for trekking.
Okay.
Okay, at that time, we carry a small amount of salt with us.
Okay.
Okay, table salt. A
Okay.
small amount of salt we are
Okay.
taking with us.
Okay.
So some leech, attack. This leech, sometime is sucking our blood.
Okay.
Okay, so at that time it's very difficult to pull the leech from our body.
Okay.
So we pour a little bit of salt on the body of salt crystal, sodium chloride. The salt crystal, a piece, we can place it on leech body. The leech completely will be dissolved.
Okay.
Salt dissolve leech body. Okay, so the same thing. Okay, if you put some salt crystal on earthworm body, earthworm will be killed.
Oh.
Completely, it will be lysed.
Okay.
Okay, so chemical fertilizer is dangerous for the worm life.
But most
Okay,
of us
so
now we are
no other
used.
way.
Yeah. Yeah. Yeah.
Yeah, that you can really understand.
Yeah.
Okay,
Yeah. Yeah.
so usually water, if you put it in salt, if you put in water,
Hmm.
salt dissolves.
Yeah.
But if you put salt on worm, worm dissolves.
Okay.
Okay, so that's the thing. Worm completely will be killed.
Okay. I
Yeah.
understood.
Any other question?
I think no questions from participants.
Yeah. Then
And now,
I
yeah,
can
yeah.
finish it.
Now I
Yeah.
request Dr. Yes
Yeah.
Chandrasekhar.
Yeah. I'll finish it. Okay.
Yeah. 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 Yeah.
Thank you all for giving me nice opportunity to share our laboratory data with you all, with all of you. I thank all the participants. I can hear, I can see Dr. Vardarajan, the Dean of Life Science, Manipur University, is with us today, and a lot more. I couldn't see all of you, but I'm so thankful for your
For
presence
your
here.
present here.
Yeah,
Yeah.
thank you. Thank you so
Thank you.
much.
Thank you so much. Dr. Chandrasekhar? Dr. Chandrasekhar?
Yes, yes, I'll
Yeah,
take care.
yeah, yeah,
Yeah,
you can,
yeah,
can.
yeah, you can. Yeah, good evening everyone to one and all here present here. Honorable Chief Guest Professor Sudhakar Subramaniam, respected Chairman, Secretary and coordinators and volunteers, all the participants. It is such honor for me to get the opportunity to thank Chief Guest on behalf of our UIT. I would like to express my gratitude to our Honorable Chief Guest, Professor Sudhakar Sivasubramaniam, to take out time from his busy schedule to grace the event. Thank you for inspiring and encouraging us with your word on this special day. I special thanks Dr. Ganesh Raja, Chairman UIT, for providing the great support to make the webinar successful. I extend my gratitude to Dr. Rajkumar, Secretary from UIT, to